Good morning, students. Myself, KK Lekha, Assistant Professor in Biomedical Engineering Department in Erode Engineering College. In today's section, we are going to see about Introduction to Pathology and Cell Injury. Okay. Well, today is our first section, so we have to know about what is pathology. Pathology is the study of the causes and effects of diseases or injury. The pathology is also refers to the study of diseases, incorporating a wide range of biology research fields and medical practices. Cell injury. Cell injury is defined as a variety of stresses a cell encounters as a result of changes in its internal and external environment. Cell injury is also known as cell damage. It is a variety of changes of stresses that a cell suffers due to external as well as internal environmental changes. The cellular responses to stresses may vary depend on the following: the type of cell and tissue involved, extent and type of cell injury. Basically, the cell damage or changes in cells may occur due to the internal and as well as external environmental changes. But now, the normal cell have a very narrow range of functions or a steady state, means it is a homeostasis. If the physiologic or a pathologic stress may force the cell to a new steady state, means it is adaptation, which means cell adapts the external stresses. If the too much stress exceeds the cell's adaptive capacity, which leads to injury, which means cell can't adapt the stresses. The flow chart shows the difference between this adaptation and cell injury. If the normal cells, which is known as homeostasis, if you are applying the stresses, the cell can adapt and changes its new position, means it is known as adaptation. If the excess stress is applied to the cells, but it cannot adapt the stress, and it is known as cell injury. Cell injury is defined in two types. There are reversible cell injury and irreversible cell injury. Reversible cell injury is known as mild and transient, which means if the injured cell can regain homeostasis and return to a morphologically normal state, which means functionally normal state, is known as reversible cell injury. In irreversible cell injury, it is a severe and progressive. It is refers to the interruption of membrane integrity. It cannot come to its normal position, normal state, normal homeostasis state. Irreversible cellular injury has two types: necrosis and apoptosis. These are cell death. The main difference between the necrosis and apoptosis is the mechanisms involved in cell death. Necrosis is the process takes place in the absence of phagocytosis. Okay, first we are going to see about reversible cell injury. It is also known as ischemia and hypoxia are the most common forms of cell injury. Reversible and irreversible cell injury produce changes in intracellular mechanisms as well as extra ultracellular changes. As already we discussed, in reversible cell injury, if the injured cell can regain homeostasis and return to a morphologically and functionally normal state. The mechanisms involved in reversible cell injuries are decreased generation of cellular ATP, intracellular lactic acidosis, damage to plasma membrane pumps, and reduced protein synthesis, which means the cellular ATP generations are formally decreased, and also intracellular lactic acidosis also affects, and also the plasma membrane pumps are damaged, and also protein synthesis are reduced. Now we are discussing the above points in deeply. The first point is decreased generation of cellular ATP. The function of ATP in cell injury is the ATP depletion or decreased ATP synthesis are frequently associated with a hypoxic or chemical injury. All living cells require chemical supply of oxygen to produce ATP. If ATP is essentially required for a variety of cellular functions such as membrane transport, protein synthesis, lipid synthesis, phospholipid metabolisms. ATP in human cell is derived from two sources. The first source is aerobic respiration or oxidation phosphorylation, which requires oxygen in the mitochondria. The second type is cells may switch over anaerobic glycolytic oxidation to maintain a constant supply of ATP. In the first supply, we doesn't need of any in the first state, we requires oxygen in the mitochondria. But in the second state, we doesn't need of oxidation. ATP is generated from a glucose or a glycogen in the absence of oxygen. Ischemia due to interruption in blood supply, hypoxia from other causes limit the supply oxygen to the cells. If there is interruption in blood supply, it is known as ischemia. The hypoxia is known as limited supply of oxygen to the cells, thus causing decreased ATP generation from a ADP. Ischemia is caused as aerobic respiration, causes severe cell injury, leads to accumulation of waste products in the cell. Ischemia affects highly specialized cells like mitochondrium, proximal tubular cells of the kidney, neurons of the CNS. Hypoxia, also known as anaerobic respirations, causes ATP generation, continues. Cell injury is less severe. In ischemia, cell injury is more severe. In hypoxia, it is less severe. Ischemia is aerobic respirations. Hypoxia is anaerobic respiration. Okay. Next, second point is intracellular lactic acidosis, mainly causes due to nuclear clumping. Due to the low oxygen supply to the cell, the aerobic respiration mitochondria fails first. This is followed by a switch to anaerobic glycolytic pathway for the requirement of energy. If this absence of oxygen, then it needs to another requirement, for an ATP generation. Results rapid depletion of glycogens, accumulation of lactic acid, lowering the intracellular pH. Fall in intracellular pH results in clumping of nuclear chromatin. Rapid depletion of glycogens results in lowering the intracellular pH. If this fall in pH level results in clumping of nuclear chromatin. Third point is damage to plasma membrane pumps. Mainly causes hydrobic swelling and other membrane changes. The lack of ATP generation interferes in the generation of phospholipids from the cellular fatty acids, which are required for a continuous repair of membranes. For the continuous repair of cell membranes, we require the phospholipids. If the lack of ATP also interferes the phospholipids generation, which results to repair of membranes reduced. This results in damage to membrane pumps operating for a regulation of sodium and calcium. Next is failure of sodium potassium pump. Plasma membrane allows active transport of sodium out of the cell and diffusion of potassium into the cell. Lowered ATP in the cell lead to increased ATPs and interface membrane regulated pump, process. The process of plasma membrane is allows the sodium out of the cell and also potassium into the cell, diffusion of potassium into the cell. If this the lowered ATP cells leads to increase the AT interface membrane regulated process. This results in intracellular accumulation of sodium and diffusion of potassium out of the cell. The process is reversed now. The accumulation of sodium in the cell leads to increase in intracellular water. If the sodium is accumulated into the cell means it leads to increased intracellular water level. Next point is failure in calcium pump. Membrane damage causes disturbance in the calcium ion exchange across the cell membrane. Excess of calcium moves to the cell, particularly in the mitochondria, causes its swelling. If the calcium is accumulated in the cell membrane excessively, which leads to cell swelling. Swelling, it affects the mitochondria and it causes the swelling. Next is reduced protein synthesis. Mainly causes the disposed ribosomes. In the potassium and sodium accumulation mainly affects the mitochondria. In this, it affects the ribosomes. As a result of continued hypoxia, membranes of endoplasmic reticulum and Golgi apparatus swells up. Hypoxia and the continuous hypoxia leads to affects the endoplasmic reticulum as well as Golgi apparatus. Then it creates swelling. Ribosomes are detached from the granular endoplasmic reticulums. Thus dispersing ribosomes in the cytoplasm and inactivating their functions. The ribosomes are dispersed from the cytoplasm and it inactivates its functions. Similarly, reduced protein synthesis occurs in Golgi apparatus, and also the protein synthesis also affected. At this point of withdrawal, acute stress is resulted. Okay, next is irreversible cell injury. In irreversible cell injury, ischemia or hypoxia results in irreversible damage to the structure and function of the cell, as we are already discussed. Irreversible cell injury means the interruption of the excessive stress interrupt the membrane, and the cell cannot come to its original normal homeostasis state. Only it leads to cell death and lead to continued depletion of proteins, reduction in intracellular pH level, and also leakage of lysosomal enzymes into the cytoplasm. Okay. Mechanisms of irreversible cell injuries are calcium influx, activated phospholipases, intracellular proteases, activated endonucleases, lysosomal hydrolytic enzymes. The calciums are inflection of calciums are acquired, and also phospholipids are activated. The proteases, intracellular proteases are affected. Then the endonucleases are activated, and also lysosomal hydrolytic enzymes also affected. The first is calcium influx. Mainly causes the mitochondrial damage. The calcium influx mainly causes the mitochondrial damage. Due to hypoxia, intracellular calcium level increases. This process disables the function of mitochondria. As a result of calcium salt on the surface of mitochondrial matrix, it disables the function of mitochondria. Then activated phospholipases. The most important event in irreversible cell injury is membrane damage. Due to the sustained ischemia, calcium level inside the cell increases, and it causes the activation of endogenous phospholipases. The calciums are sedimented in the cells, then it leads to endogenous phospholiposis activation. Finally, the result is damage to lipid bilayer membrane and reduction of ATP generation. Then the next process going to happen is intracellular proteases. It mainly causes cytoskeletal damage. The normal cytoskeleton of the membrane is damaged due to the activation of intracellular proteases, are by physical effect of cell swelling produced due to the irreversible cell membrane injury. Irreversible cell injury damages the cell membrane, then it leads to cell swelling, and the intracellular proteases is activated. It affects the cytoskeletal damage. The next process is activated endonucleases. It is mainly caused due to the nuclear damages. The nucleoproteins are damaged by the activated lysosomal enzymes such as proteases and endonucleases. If the proteases and endonucleases are activated, the lysosomal enzymes are activated, and the nucleoproteins are damaged. The next process is lysosomal hydrolytic enzymes. Mainly causes lysosomal damage, cell death, and phagocytosis. The next process is lysosomal hydrolytic enzymes. It mainly causes the lysosomal damage, cell death, and also phagocytosis. The lysosomal membranes are damaged, and the result in escape of lysosomal hydrolytic enzymes. If the membranes are damaged, the hydrolytic enzymes are came out from the membrane cells. These enzymes are activated due to the lack of oxygen in cell and acidic pH. The lack of denaturation of ATP leads to lack of activation of enzymes. Hydrolytic enzymes include hydrolase, RNase, DNase, protease, glycosidase, phosphatase, lipase, amylase, etc. These enzymes are activated, which leads to digestion of cellular components. The cellular components are digested by the enzymes. This results in cell death. The death is eventually replaced by a masses of phospholipids called myelin figures, which are either phagocytosis by macrophages or other may be formation of calcium. Either the formation of calcium or activation of enzymes, the results which results in cell death. The below figure shows the ultrastructural changes during the cell injury due to the hypoxia and ischemia. The first is normal organelles of the cell, and the next is defined reversible cell injury, and the last column is defined irreversible cell injury. In this first, the normal mitochondria is seems like normal. In this reversible cell injury, the amorphous densities are produced and also cell swelling, mitochondrial swelling is produced. If this amorphous density is larger density is more larger, and also the swelling with vascles also produced in the irreversible cell injury. The normal membrane in irreversible cell injury, the blebs, small blebs, and also membrane particles are produced, and also cell swelling also produced. Myelin figures also formed in a irreversible cell injury. But in irreversible cell injury, the myelin figures and also disruption of the membrane produced. In rare and ribosomes, in irreversible cell injury, the cell swelling, ribosome swelling, and also dispersed ribosomes are appeared. In irreversible cell injury, the lysed ribosomes and dispersed ribosomes are produced. In lysosomes, the reversible cell injury leads to autophagy. But in irreversible cell injury, the swollen and ruptured lysosomes are appeared. In cytoskeleton, the aggregated cytoskeleton are produced in reversible cell injury. But in irreversible cell injury, the disrupted cell injury are produced. In nucleus, clumped chromatins are formed in reversible cell injury, but in pyconosis and karyohexis are produced in irreversible cell injury. This shows the how the changes in reversible and irreversible cell injury in cells may happen due to the cell injury. The difference between the images of the mitochondria, membrane, ribosomes, lysosomes, how it seems in reversible cell injury and irreversible cell injuries seems in this picture. Next, we are going to see about the differences between the reversible and irreversible cell injury. The main difference is the morphological and structural changes, as we discussed in above. In reversible cell injury, it is a type of cell injury that can return to the steady state, which means normal homeostasis, by altering cellular conditions. The cell structure changed, but it can comes to its normal state. But in irreversible cell injury, one of the severe type of cell injury which leads to cell death, cell can return to its original position, normal position in reversible cell injury. But in irreversible cell injury, it cannot be returned to its original position. It has passed the point of no return. In reversible cell injury, sublethal and short acting. But in irreversible, it is lethal and also long lasting. Reversible cell injury is caused by a lack of oxygen such as hypoxia or ischemia or blood flow to the cell. But in irreversible cell injury, it is caused by immunological responses or viral infections. Reversible cell injury results in cellular swelling and fat accumulation. But in irreversible cell injury, it is results in necrosis and apoptosis. Reversible cell injury can be treated with drugs, but in irreversible cell injury it cannot be treated. It leads to permanent cell loss. Okay, as we discussed in above, irreversible cell injury leads to cell death. Cell death has mainly two types. It is an irreversible cell injury, changes in the cell associated with its end result in different types of cell death. The different types of cell death are necrosis and apoptosis. Mainly necrosis is a cell death of body tissue. It occurs when too little blood flows to the tissue. If the cell receives so much of low blood flows means it is known as necrosis. This can be formed from injury, radiation or chemicals. Necrosis cannot be reversed, which means it cannot be come to its normal state, homeostasis. When the large area of tissue die due to lack of blood, necrosis also plays a role in preventing cancers. The flowchart shows the normal cells and the difference between necrosis and apoptosis. The normal cell's nucleus and organelles are displayed normal, but in the necrosis, the small blobs are formed. The outside the layer of the cell is membrane of the cell is formed as a small blobs. The structure of the nucleus also changed. The blobs fuse and become larger. In the second stage, the blobs are fused and also it becomes larger. No organelles are located in the blobs. The organelles are not located in the blobs. In the final stage, the cell membrane ruptures and releases the cell's content. The cell membranes are ruptured and it releases the cell contents. Nucleus and organelles are releases from the cells. The organelles are not functionals. In apoptosis, also same like small blobs are formed and it leads to the nucleus begins to break apart. Then it is a programmed cell death. So nucleus begins to break and the DNA breaks into a small parts. The nucleus and also organelles are breaked into small pieces. The organelles also located in the blobs. But in necrosis, the organelles are not located in the blobs. And finally, the cell breaks into several apoptotic bodies, the organelles and functionals. And finally, the cell breaks into several apoptotic bodies. The organelles and are still functional. But in necrosis, the organelles are not functional. In necrosis, the cell membranes are ruptures and it releases the cell contents. But in apoptosis, their cells are divided into smaller bodies, not of several bodies. Okay. First, we are going to see about necrosis. As we said already, it is an accidental or ordinary cell death. It is a pathological cell death which occurs when cells are exposed to a serious physical or a chemical insult such as hypoxia, hyperthermia, ischemia. Necrosis is the sum of morphologic changes that follow cell death in living tissue or organ. There are mainly two process influences the necrosis: enzymatic digestion of the cell and denaturation of the proteins. And the necrosis are the programmed cell death. It is mainly caused due to the lack of blood and oxygen to the tissue. Examples of necrosis are hypoxia, ischemia, and hyperthermia. The enzymatic digestion of the cells have two types. First is autolysis, the digestion of cell by enzymes derived from their own lysosomes. The next is heterolysis, the digestion of cell by the enzymes derived from the lysosomes of leukocytes. If the digestion of enzymes is derived from own cells, means it is autolysis. If the digestion of cells by enzymes are derived from lysosomes of leukocytes, is known as heterolysis. Denaturation proteins caused by a intracellular acidosis results in injury to the cell membrane, severe impairment of phosphorylation of cell, increased permeability of the cell, influx of Na plus and Ca plus in the cell, and also intracellular activity of the cells decreased. Dead cell morphology. In cytoplasm, there is an increase in eosinophilia and glassy appearance of cytoplasm due to the loss of glycogen particles. Swollen mitochondria. The mitochondria is swelled. In nucleus, the clumping of chromatins are formed. In pycnosis, nucleus progressively shrinks and becomes dense moss and tightly packed of chromatins. In charolysis, progressive dissolution of nuclear chromatin due to the action of DNase and lysosomal act again. The causes of necrosis are may be acquired due to the external and internal factors. The external factors are mainly mechanical trauma, damage to blood vessels, thermal effects. Mechanical traumas are like physical damage to the body that causes cellular breakdown. In damage to the blood vessels, which may disrupt the blood supply to associated tissues. Thermal effect, extremely high or low temperature results in necrosis. The internal factors are trophoneurotic disorders, pancreatic enzymes, immunological barriers, bacterial toxins, toxins such as venoms. The trophoneurologic disorders are injury and paralysis of the nerve cells. The pancreatic enzymes are major cause of fat necrosis. The immunological barriers, invasion of pathogens through surfaces affected by the inflammations. the bacterial toxins and also toxins such as venoms. The types of necrosis are coagulative necrosis, liquefative necrosis, caseous necrosis, fat necrosis, fibrinoid necrosis, gangrenous necrosis. The first one is coagulative necrosis. In this type of necrosis, the necrotic cell retain its cellular outline for several days. The coagulative necrosis typically occurs in solid organs such as kidney, heart, adrenal gland, except brain, usually as a result of deficient blood supply. For example, mitochondrial infarction. The gross appearance of coagulative necrosis are necrotic area is firm, opaque, and with cooked meat appearance. In microscopic appearance, the architectural outlines are present, cellular details are lacking. Results in dead tissues remain in the body for a long period, ultimately removed by macrophages. In liquefative necrosis, it is a transformation of the tissue into the liquid viscous mass. The affected cell is completely digested by the hydrolytic enzymes, resulting in pus and fluid formation in the tissue. For example, brain infarction. In cross appearance, the necrotic tissue is liquefative and filled with semi-solid creamy liquid called pus. Pus is a thick white or a yellow liquid consists of exudate of leukocytes, tissue debris, and microorganisms. Microscopic appearance, it is shows like no architectural or cellular details are visible, but in necrotic area appears as a cavity containing mass of necrotic neutrophils and bacteria cell debris. Necrotic area appears as a cavity containing mass of necrotic neutrophils, bacteria, and cell debris. The next is caseous necrosis. It is a uniform cell death in which tissue remains a cheese-like appearance. It is called distinctive form of coagulative necrosis. For example, necrosis in lung and kidney. In gross appearance, it looks like white, soft, cheesy looking material. In microscopic appearance, it looks like the necrotic tissue is amorphous, granular mass enclosed inside the zone of granulomatous, inflammation containing macrophages. No architectural details are seen. The next type of necrosis is fat necrosis. Death of adipose tissue in a living animal is known as fat necrosis. There are three types of fat necrosis: pancreatic necrosis, traumatic necrosis, nutritional necrosis. In pancreatic, death of adipose tissue in and around pancreas. In traumatic, it is a subcutaneous necrosis caused by mechanical injury. In nutritional, it is mainly seen in a sheep with extreme debility and emaciation. In gross appearance, necrotic fat appears as a white or yellowish chalky masses. In microscopic appearance, the necrotic tissue is solid and homogeneous. Next type of necrosis is fibrinoid necrosis. It is a type of necrosis occur in a connective tissues, especially affecting arterial walls. For example, autoimmune diseases. The example of autoimmune disease is rheumatoid arthritis. It is characterized by loss of normal structure of collagen fibers. The next type is gangrenous necrosis. It is an extensive tissue necrosis caused by bacterial infections. Three types of gangrenous necrosis are dry. It is skin look black and dry, lymph losses, blood supply, and coagulative necrosis. In wet, the bacterial infection superimposed liquidative necrosis. Tissue is swollen up, reddish black with extensive liquefaction. Gas, wound infection, presence of gas in tissues. These are the pictures shown the types of necrosis. The coagulative necrosis in microscopic view, the liquefative necrosis, and gangrenous necrosis in a leg and foot necrosis and fibroid necrosis. The next is apoptosis. As we know, the apoptosis is a programmed cell death, normal or a programmed cell death. The apoptosis is physiological cell death in which unwanted or a useless cell are eliminated during the development or the normal biological process. It is important for development of multicellular organisms, homeostasis of tissues. The causes of apoptosis are physiologically, the causes are embryogenesis, hormone dependent evolution, immune response, cell death induced by cytotoxic T cells. In pathology, the causes are by injurious stimuli, viral diseases, atrophy, tumors. The morphological changes in the apoptosis are cell shrinkage, organelle reduction, mitochondrial leakage, chromatin condensation, nuclear fragmentation, membrane blurring, and changes. This figure shows the apoptosis flowcharts, how the apoptosis happened, how the programmed cell death is happened. In normal cell, the cell shrinkage and chromatin condensation are formed. Then it leads to membrane blurring. The blurring of the membrane is formed. Then the nuclear the nucleus are collapsed and continuous blurring is happened. Then it goes to apoptotic body formation, which means the cell is differentiated in separated as the several bodies. Lysis of apoptotic bodies are happened finally. There are two types of pathways for apoptosis. There are intrinsic pathway, extrinsic pathway. Intrinsic pathway is nothing but the activation of apoptosis via mitochondria. Inside the cell, the apoptosis process is happened. As an intrinsic pathway, when the stress signals, DNA damage signals, and defect in signaling pathway are processed. The intrinsic pathway uses mitochondria as a central component or the activation of apoptosis. In this pathway, the intracellular cells leads to activate the pro-apoptotic proteins BACs with releases of cytochrome C from mitochondria. Formulation of the apoptosis and activation of initiator Caspase-9 happened. The activation of Caspase-9 finally executioner caspases are activated and the cells are destructed by proteolysis. The apoptosis via the pathway can be controlled by various anti-apoptotic proteins including BCL2 and inhibitors of the apoptosis. These flowcharts show the intrinsic pathway of the apoptosis process. How the Noxa and Puma back and backs are uh, supplied, and the mitochondrial cytochrome and apoptosis are happened. How the caspase 9 is released, and also caspases 3 and 7 are released. Finally, the apoptosis are happened. An extrinsic pathway uses extracellular death ligands like Fas ligand, tumor necrosis factors, TNF, to activate cell receptors which pass the apoptotic signal to initiator caspases. Initiator caspases and also executioner caspases. The initiator caspases are examples caspase 8, but in executioner caspases, examples are caspase 3 and 7. In the execution phase of apoptosis, various cellular substrates are degraded, leading to cellular collapse. As a consequence of caspase activation, the number of key enzymes and structural proteins of the cell are degraded, leading to cell death. The figure shows the cell death of apoptosis extrinsic pathway. Cell extrinsic pathway, the pro-apoptotic ligand and death cells are initiated. Caspase 8 and caspase 3, 6 and 7 are initiated. How the apoptosis is processed. Caspases are central medium to apoptotic program. Caspases and 8 and 9 are initiator caspases. Caspases 3, 6 and 7 are executioner caspases. Family of BCL proteins, gatekeepers for apoptosis. The BCL2 proteins are gatekeeper for apoptosis process. Group 1 anti-apoptotic proteins, A1, BCL2, BCLW, BCLXL, MCL1. The group 2 are pre-apoptotic proteins like PAC and BAX. Group 3 proteins are BIT, BAD, BIM, NOXA and Puma. The tabular column shows the differences between the necrosis and apoptosis. In the first is size. The cellular swelling is happened in necrosis. The necrosis is uncontrolled cell death. Apoptosis is programmed cell suicide. In apoptosis, cellular shrinkage is happened. Many cells are affected in necrosis, but only one cell is affected in apoptosis. In uptake, the cell contents ingested by macrophages. In necrosis, in apoptosis, cell contents ingested by neighboring cells. Significant inflammation is happened in necrosis. No inflammatory response is happened in apoptosis. In membrane, loss of membrane integrity is happened in necrosis. The membrane blubbing and but integrity is maintained in apoptosis. Cell lysis occurs in necrosis. Apoptotic bodies are formed in apoptosis. In organelles, the organelle swelling and lysosomal leakage are excreted in necrosis. In apoptosis, mitochondria release pro-apoptotic proteins. Random degradation of DNA is happened in necrosis. The chromatin condensation and non-random DNA degradation is appeared in apoptosis. These are all the changes that happen in necrosis and apoptosis in size, uptake, membrane and organelles of the cell. Thank you for listening my session.